Hi.
Hi, how are you?
I don't know. I have to give this, it's only half an hour
Okay,
speech.
all right.
It's 40 pages, 40 slides long, but 7 of them are just sort of plain titles, so it's more or less 30 pages.
Yeah.
And for some reason, it's giving me a bad time. But anyways, I can't really spit out some of the things that I need to say. So
No.
it's, would you like to see it or
Yeah.
some of it?
Yeah, some of it would be great.
We'll try this. Can
There we
you
go.
see that?
Yes, yes, I can see it. You have to
Well, the entire
translate.
screen, that aspect. Okay. And I'm supposed to be giving it like this, but somehow it cuts off University of Manitoba, December 2024. But anyways,
Oh,
um, anyways, the title of the whole thing is modeling simple epithelium tissue using tensegrity structures. And you can tell I just not with this. You and I are not one in accord. Okay, so I have to. Works there. Okay, so my outline. Insist that I have an outline. So introduction, epithelial tissue structure, model build results, validation, and conclusion. Okay, so this is the introduction. Epithelial tissue structure. Epithelial tissue comes in several different types, squamous or short, cuboidal and columnar, which is tall, stratified and pseudostratified, and my models are squamous, cuboidal, and columnar. And biological tissue is a structure in tension, and it is considered to be a tensegrity structure. So this is a stress-strain curve of skin. And you can see that when it's relaxed, it has an elasticity that is kind of flat. And when it is tensioned, it changes to a larger elasticity when the structures are aligned inside. And then this is when it breaks. So curves like this are going to be used for the validation portion of my talk. And okay, epithelial tissue has an actin belt structure at the top. It goes around in a belt around the upper part of the cell. And it also has a basement membrane, and it's attached to the basement membrane. So these are showing an actin and myosin stress fibers that make up the belt. And it's non-muscle myosin, but it still acts like muscle does and contract. So this is where you get the tension in your structure, in your epithelial structure. And trijunctions in cells are between the cells. You can see them here in yellow. And here in blue, they occur at the vertices in the cells. And this is a close-up view of one, or close-up diagram. So epithelial tissue structure has a top ring. See the top ring here?
yep.
And then it has trijunctions, and they're quite small. Trijunctions here and here and here. And then it has basement membrane attachments all along in blue here. And tensegrity models are made up of strings in tension and bars in compression. And the whole structure is tensioned. Oops. Oh, good. It works with, so it works with my scroll bar on my mouse. Anyway, so my model build. This is a T3 tensegrity in tension, showing it being pulled in. And this is a T3 being compressed. And these were used for the tri-junctions with the posts. Show you the posts in a minute. And this is a T4 tensegrity, again showing tension and compression with the stress shown in colors. So as it becomes tension, there's more stress on the strings. These are the strings here, and these are the bars. And same here, these are the strings, and these are the bars, and the whole thing is in tension. So I made this out of hexagon tilings, and the bottom of the hexagon has the measures of S, K, and W, and then M. And M is the membrane distance between the cell membrane.
Okay.
Cell membrane. And then these are the building blocks. This plane attachment didn't work at all. So I made tensegrity attachments with the strings, and in blue in this case, and the black in black is a bar. And when you place these tensegrity structures in tension, they tend to curve a bit, and the same with compression. And this is a hexagon 3D structure. I made it out of 2 hexagon rings, a smaller one at the top, larger one at the bottom. And the compression, the compression elements were added in blue. So these are the bars in blue on the top and bottom. And then this is the 3-cell structure. And in the middle, I have this T4 structure, and attached to these posts, I have the T3 structure, and the posts are necessary to hold the structure up, and they can represent the rest of the tissue, since this is only 3 cells. And here's another view of it. You can see that the T4 structure is quite small, and the T3 structure is as well. And I made 3 basic models: a cuboidal model, a squamish or short model, and a columnar or tall model. They all have a spring foundation that you can see in blue. So these are springs. And I chose a variety of spring constants from 10,000 newtons per meter to 10 newtons per meter. And that's quite a large range, but tissue also has a large range of elasticity. Let's see. Oh, I didn't include the range of elasticity for tissue. I need to find it. And in order to get the structure to work, I had to have pin points. So these are the pin points on the outer part of the posts. Anyways, so then I used pre-strains from 0 to 0.3, and this is for the range in tissue it's normally used. And forces between 0.1 nanometers and 200 nanometers. And this is a range for small tissues samples taken from atomic force microscopy. There's the forces. They were applied to the middle of the structure on top of the T4 structure in the middle, and there's the T4, and here are how the forces are applied. And for engineering stress, I simply used the area of this triangle, and here's the calculations for the triangle that's on top of the T4 structure. For engineering strain, I used the original height of the cells. So the Squamish model was 1.2 micrometers high. The cuboidal model was 3.5 micrometers high, and the columnar model was 20.5 micrometers high. And so engineering strain is a change in height divided by the initial model height. So this is an overall view of what I did for my experiments. I made hexagon 3 model with the Squamish, cuboidal, and columnar wide posts and columnar thin posts. I tried some brief experiments modifying the top and bottom of the structure, and I changed the forces applied to the structure, I changed the pre-strain, and I changed the spring foundation. Results. It was found that the 3-cell tissue model mechanics were influenced by the cytoskeleton values, or the string, I guess I should say string and bar values, and the cell-to-cell connections and the cell-to-basement attachments. And this occurs with other models and with the measurement tissue. They are reliant on their attachments and their cytoskeleton values. And these are sample graphs, and this one is in tension, and this one is in compression. And this is going with the convention where tension is going negative and compression is positive. That's just the reverse convention. And this shows the different pre-strains that I tested. And you can see a good separation of curves in both tension and compression. And mostly this separation of curves for both tension and compression occurred in the Squamish model, not the other models. And then there's a K value that is the area of the string, cross-sectional area of the string, times its elasticity divided by the cross-sectional area of the bar or rod. I guess it's a bar, 29 bar. And to give you a K value, and this actually changes the slope of the elasticity curve. And for my model, I kept the truss posts with the same values as the bars. And the truss post, the elasticity is both in tension and compression. So the truss post is not a tensegrity, it's a twist. So this is showing what happens with the cell-to-cell connections. So if I have a larger distance between cells, the membrane distance of 0.12 micrometers, you get this type of curve, and it's further apart, and you get good separation of the curves. But if you have a separation of 0.05 micrometers, then the curves are all on top of one another and become linear. And cell to basement membrane attachments, if you change the spring foundations, sometimes you'll get a curve with good separation, like this one, with the columnar wide model, but mostly this didn't happen. So I removed the middle attachment, spring attachment to the base of the model, and tried this with the Squamish model, and the curves basically collapsed and became very nonlinear here. So this is perhaps a disease state when you have the middle completely vanished. And it's more, I guess, detaching a spring foundation is more important than changing its spring constant. So validation, someone, you et al. Made a really nice experiment where they aligned cells on a single sheet. So they oriented the cells. So the oriented cell sheets are here, and this is a non-oriented one. And my tensegrity model curves can match these oriented cell sheets, at least for this one, oriented cell sheets -15, so 15 micrometers by 15 micrometers. And for this one, I get a very close match with the oriented cell sheets -5 when they're tensioned and perpendicular with their alignment. And that happens mostly with the pre-strain 0.1 is a very close match to the oriented cell sheet -5. And then this is in compression. This is the original graph. And what I have here are the average of all of these measurements. And then this is my model, and it's a little high compared to the lung tissue here, but it does match within, I don't know, 30%. And the interesting thing about this is the curves are reversed. The 45% pre-strain is lower than the 0.1% pre-strain, and normally that doesn't happen. Normally the high pre-strain is the higher elasticity curve, and the lower pre-strain is below it. But my model does model this reversal in some instances. And then I tried this with human skin just to see what would work. And the Squamish model came in here, and the cuboidal model is here. And the cuboidal model, we're saying, sort of matches the young skin, but the Squamish model kind of does too. And this is older skin on the bottom. So there is somewhat of a match. And if I worked on this more, I could only get it to match better, get a slightly different model. Conclusions. So my hexagon 3-cell model can model tissue. That's a mouthful. And actually it's more linear than the the models of tissue quite often. But there are only 3 cells in the model. So perhaps a model with more cells in it would work differently. The main conclusion is that the tissues and the model alike are reliant on their attachments and values of its structure, as in the elasticity of its strings and bars. I keep interchanging bars and rods. Anyway, and the hexagon 3-cell model can handle large stress and strain. Normally, models that are used for tissue are up to 0.2 or 20% strain or deformation, and most tissues yield at around 50% strain, and then therefore my model is good for modeling tissue. Questions? And I just don't know.
Well,
Anyway,
that was very good. Thank you. Yeah.
Okay. Any comments?
I'll bring your slides back up for a minute.
Oh, okay, sure. Ill coordinated.
Okay. Yeah, it looks like Dick's here, so I don't know if he wants to ask questions, but I had a few questions. So yeah, go back up. So I guess one thing, like when you go to the beginning of the talk and you go through and you start introducing, yeah, like you talk about epithelial tissue, then you go down to the next one. Yeah, go to the next one. That's a nice curve. That's actually like you're showing the different types of like tissues, and then you're showing like damaged tissue, and then the curve drops off.
So just a skin sample and what happens to it when you tension it, and when you tension it too much.
Oh, okay.
It breaks. Yeah. So this is just a skin sample, and it starts out relaxed with a low elasticity, and then it tensions, and the structures inside become more aligned.
Okay.
Okay, so I need to explain that better.
Yeah.
Skin sample. Actually, if I get through it without tripping over my tongue, I'll be just pleased to answer any questions.
Yeah.
So,
good. Yeah.
So what else would you like to see here?
Then you kind of jump into tensegrity. So like if you go down maybe another couple slides.
Yeah, I think you were kind of.
Yeah, so you get into the tensegrity model. You don't really talk about tensegrity much. Like you don't motivate the tensegrity. I don't know if they're used to hearing about it in your group or not, but
No, I should be explaining it, but
yeah.
I have 30 minutes and 40 slides. So basically a tensegrity is strings in tension and bars in compression, and they're held upright by being in tension. They're a structure that's in tension.
Yeah. Okay. So I mean, yeah,
And
you
they're
probably
different from any other mechanics. But anyways.
yeah. So
what else should I have something?
Well, I mean, I don't know. Yeah, given your time constraints, I mean, I would put in like maybe one highly visual slide that shows like how they have tensegrity structures, like mechanical tensegrity structures, like geodesic domes or things like that, and showing like that it's something that is ultra stable. So there are examples of tensegrity. You have geodesic domes, you have buckyballs, you have things like that that are at least models, if not outright things that you can demonstrate the ultra stability.
Well, and what I do have is I have my tensegrity structure with me.
Okay,
I
yeah. So
could,
you have the model. Yeah, the
so
physical
I
model.
could actually hold it up and say, here it is. This is
Yeah.
a tensegrity structure. And I probably will. And
Okay,
just
yeah.
in the, this is it. I'll just do
A picture.
a real life model visual when
Yeah,
I'm,
yeah. That would be good.
yeah.
That would be good.
And then this is a very common tensegrity structure, this T3. They
Okay.
use this all the time. And I just got a paper off the internet that says that they use this tensegrity structure to model E. coli and looked at how it sticks to surfaces.
Okay.
Anyway, it's about the simplest 3D tensegrity structure out there.
Yeah. So what are E3 and E4? What do those stand for? Or T3 and T4, I'm sorry.
T3 has 3 bars in it,
Okay.
and that's how they named that. And then this is the T4, and I don't know why they called it a T4, because it's just two T3s on top of each other. They're mirrored, so they have a more linear movement than the T3s.
Oh,
So they're good structured for the middle of my tensegrity because they're kind of linear. They don't uh, twist too much
yeah.
with in tension and compression. Anyways, this is showing the stress in colors.
Yeah.
So the blue is less stressed and the red is very stressed. So it was mostly the strings in this model taking the stress and strain.
Okay. Yeah, that's cool. I mean, like you can see the simulation at least, like how it deforms and the change in the values.
Yeah, I wanted to do that for the hexagon model, but I would have had to put together a movie, and that might not have worked either. I had to feed it individual forces in order to get it to work, and these structures, probably because they're a triangle, worked fine with their automatic changing of forces between two limits, but
Okay.
not the other one. You go down here and you build this thing, and this barely stands up by itself. And when you build this, the only way I could get this to work was the T4 in the middle and the T3s on the outside attached to the posts. I just discovered that one day, or I wouldn't have a project.
Yeah, yeah.
Will not stand up if you put the T3s or T4s, I mean, on the outside. It won't. Very fussy. These tensegrities are very fussy objects. Maybe because the world thinks in compression.
Yeah.
Yeah. And then
So the
they have the,
trijunctions. So you had the trijunction here, and then you had, so the trijunctions are kind of in the middle of the structure. And like, it doesn't matter if you go in like, the trijunctions are just like the junctions and in the same plane. Like when you have the attachment, that doesn't count. As I guess it doesn't
yeah,
count as a junction.
this is the only attachment that's attaching these three together in the middle.
Okay.
And it's only for the top part of the tensegrity structure. It just attaches to the top. That's how I can see it in this one. It just attaches here to that top ring, to this top ring.
Yeah.
and nowhere else. Like this, these are not attached. The sides of the cells here are not attached. In real life, there are other structures there, but in reading it all, they say that it doesn't come into play until the cell is like 80% compressed or 70%. 70%.
All right. So why
So
is it a T4, this structure?
why is the T4 here?
Yeah, why is it a T4 instead of a T3?
Because the structure does not work and falls apart if I put a T3 in the middle.
Okay.
I don't know why.
Huh.
So if they want me to do a PhD on that, I guess I could look into it, but I really don't think I want to.
Yeah, that's the way it goes. Every slide is a PhD, so potential
Yes,
PhD.
yes. This is like, why does this work this way?
Yeah.
Yeah, question. I don't know. I just discovered this one day.
Oh wow. Yeah.
I kind of knew I needed a T4 in the middle because it's a very stable, it's one of the most stable tensegrity structures out there. And it doesn't twist, and I could attach it to the top and bottom of the top ring here.
Okay.
Any other nice questions? And
Well, just
no,
keep going through the slides, and then Dick has a couple questions. But yeah.
well, I have. No, that's not. It's 27 where I made the mistake.
Oh.
Okay,
What was the
so
mistake?
cytoskeleton values. The string and bar values.
Oh, okay. So it's the model, not the biology.
Yeah, trying to do this.
Okay.
Because they don't know, my audience or engineers might not know what I mean by a cytoskeleton.
Oh yeah.
And this is not a rod, this is if I can, put a bar in here.
Yeah.
There. I've been trying to call them bars, and it just seems to be interchangeable sometimes in the literature.
Yeah.
Okay, so, Nick, did you have any questions?
You had two in the chat.
Oh, okay.
Three. Well, he said Beth Burnside showed axial microtubules. So that's
Yeah, so if they want to know what part of the cytoskeleton I mean, then I'll just tell them that. I did explain it in my thesis. The microtubules have a larger, what do you call it? Continuation length? What do you call that? Anyway, they are long enough or occur at a long enough distance to be bars in the cell. And then I'm taking Dick's famous, and I guess he said somebody else showed this to him, a dowel, a long dowel, and a bunch of elastics. And if they want to know why I made my bars the same value, elasticity value as steel, I can show them how that works. Because microtubules are actually around about a gigapascal or 2 gigapascals elasticity. So that's not 200. So how does it get to be 200? Well, you take your dowel and you put elastics up and down it, and if you tension the elastics, it becomes stiffer. And you can just demonstrate that to the audience. So I'm going to use it like they're going to ask this question, and I'm going to get Dr. Zhang to hold the dowel up because it's tension, and there's actomyosin throughout the cell. It's just not in it, most of it's concentrated in the belt at the top, but it's also distributed throughout the cell and is attached to microtubules. I just saw a paper on it. I didn't quote it. If they want me to quote all of my sources and the things that I'm saying off the top of my head, I could have like 500 references in this paper.
Yeah.
Oh, how much computer time for 9 cells? Well, 7 cells would give you a nicer model, in my opinion. This computer, and this is a 6-year-old computer, will do this in like 2 seconds, the 3-cell model. So yeah, it'll only take like seconds, probably. Anyway, Rodland and Gordon. Oh, yes, and I quoted someone who says that at the 70% strain, which most tissues apparently don't get to because they break before that, well, at least the dead tissue. Well, even the live tissue there, it was only getting up to 60%. But anyway, if it gets to 70%, the vinculum strings are attached, and they prevent buckling. Yeah. Okay. Then the intermediate filaments. Yeah, intermediate filaments are attached to the nucleus and attached to the desmosomes on the side of the cells. And there's also, I just got a paper and says there's punctum along the side of the cells as well. So I haven't included intermediate filaments, and I have said that the attachments are at the top with the trijunctions, the T4s and the T3s. And that was my very simple model. This is the simplest model I could make of tissue. 3 cells and about 4 attachments at the top belt. Anyway, you can't unmute to hear more questions.
I mean, yeah, if you had more questions, put them in the chat. But
okay, and please don't post this until my thesis is handed in somehow. Right. Because, okay. Because discovering that it needed a T3 in the middle like that is my thesis, or makes my thesis, because then I was able to get a model to work, and then I was able to test it and see that there was good separation of curves, or see what conditions caused good separation of those curves, and where it was nonlinear. And where it's nonlinear, I think it could produce a J-curve under the right conditions. This is wobbles around.
Yeah.
I don't know how to say that. And anyway, it's got a nonlinear portion, and then it becomes linear as it gets stretched. So,
right.
Yeah, and I'm near Winnipeg in Niverville right now, and it looks like it's sleeting or something.
Oh, okay.
Yeah, so I'm close to Winnipeg, so I'll hopefully be able to drive.
When is your defense? Or...
At 3 o'clock this afternoon.
Oh, okay. Good luck. Yeah.
Anyway. Yeah, 5 years work. Please explain it in 30 minutes. I still have my slides here if you have another question.
Okay. Well, yeah. Let's go through the rest of your slides. It would be.
I'm aware of microtubules being long and being the bars in the situation. I don't know if the engineers care. So you want to see the slides again?
Yeah. Yeah. Let's do that.
Okay. Share screen. Where's the share screen? No, I'm sharing screen. Okay.
Yeah. You
And
have to pull up your PowerPoint.
yeah, so can you see that?
Yeah. Yeah.
You can see my cytoskeleton values.
Yes. Yeah.
Cytoskeleton and again, I need to do this. String and bar.
Oh,
And in,
cytoskeleton.
I'll just put that in brackets so I can introduce that to them if they need it introduced.
Yeah. That's good. Oh, that's nice.
Yeah, and this was a nice find as well because you can adjust this K value and it changes the curve on my model. Mind you, I've got it down so that it only withstands about 2 nanonewtons of force because it's so small. But if you're using atomic force microscopy, that's what you get. Like it's small, like it was being measured with atomic force microscopy. If you make the strings, this 0.5 megapascals, as though it was elastin, that's the value of elastin in a cell. So I guess it was Stephen Levin said, don't make a squish. He says that you shouldn't make your strings elastic. Well, I think some cells are squishes. So that's one of my conclusions from all this is that some cells are squishes. So I don't know whether I should tell my committee that. So there's a very broad range of elasticity here, and that's because tissue has a broad range of elasticity. So I made it like a broad range, of course, to try this out. And more or less, I'm trying this out within what I consider to be a living cells range. So most of my models that I match to the tissue are K equals 5 times 10 to the minus 9.
Oh, yeah, okay.
Yeah, which don't really match the forces in anything except that single cell sheet. But this single cell sheet here is closer to my model because it's 3 cells and these are like, how many? 50?
Yeah.
I don't, so.
Okay.
So any other questions? Can you say them out loud since I can't see the text? Dick, do you have questions that Bradley can reinterpret or? Anyway, I was very pleased this one worked so well. Like I said, it's 5 times 10 to the minus 9. Squamish cell matches this. And these cells, these oriented cell sheets, were made out of cardiomyocytes. So, I don't know.
So when you say a perpendicular force, you mean like something that kind of comes from the top or from the sides?
Side to side, because these are oriented cell sheets, so they've oriented them in groups. So they're aligned. You can see it in the picture actually. So these OC5s, -5s are aligned along these groups.
Okay.
So this graph here is tensioning them in the aligned direction, and these ones here are perpendicular to the aligned direction, so they're the other way.
Okay. So it's like it's the perpendicular to the alignment.
Yeah, so it's a perpendicular stretch. So it's stretching, the perpendicular stretch is stretching this way, and this is an aligned stretch, so it's stretching it this way. I have a picture of their homemade device where they glued these cells and then stretched them, and they've made a similar type of clip arrangement for measuring cardiomyocytes. So they were talking to each other. They got cardiomyocytes from the lab that was doing this, and they made a single cell sheet and oriented cell sheets. It's kind of interesting.
Yeah. Dick has a question. If the cell contracts, will an adjacent cell also contract?
In the cardiomyocytes, they show it that when they pull on it and it contracts, and pull on it and it contracts. I haven't really looked at that, but there is, if you go into this paper, they're thanking the lab for the cardiomyocytes. I think we could probably go back from that, but they've been studying cardiac tissue. So if you want to answer that question, I guess look at some of the cardiac stuff. They've also been looking at lung, but lung is complicated. Like this is a cadaver. I think it's pig lung. But it's a complicated tissue. There's squamous cells mixed in with some ligament stuff.
Yeah. So yeah, that's the other question is like, if you have a diversity of different sort of arrangements and you apply a force and they're kind of in there together, what does that look like? I know that's kind of a lot harder to model.
Yeah, so yes. So this isn't an exact model of this particular tissue.
Yeah.
Like I said, the cell sheets are much better. But they didn't do the cell sheets in compression, they just did them in tension. And so this was actually a thesis. You can see this. Devin Wozniak did this, and he said he did it because there was a lack of data in the literature. Yeah, there's a lack of data, especially for compression. So we compress lung tissue. Yeah, and then basically there was a lack of data. There weren't very many graphs for me to compare this with. A lot of them do time studies where you're giving pulses of force to their tissue. Mind you, atomic force microscopy is in compression, but it's they're measuring one cell. Like in my thesis, I have one example where they're measuring the force between two cells. They might be pulling on that. Actually, I don't. Anyway, it's in my thesis.
Good.
And these, like, they're pulling on these, and my model is actually compression and tension, both compression and tension. So this is so I use the tension. These are from my model, and they're tension curves. Same with this one. That's a tension curve. And then I was using compression curves here. These are compression curves. like I said, there's this inverse where you've got the pre-strain, large pre-strain is the bottom curve. Normally it would be the top, and I could get my model to do that only in a few instances. So this is a very particular law.
Yeah.
I guess it's 8 times e to the minus 9, the k value of spring foundation of 1,000 newtons per meter in compression. And I compressed it the same with the same pre-strains they did. So I did 0.1, 0.2, 0.35, and 0.45. And I got a result. But if you change the amount, like if you made that 10,000 newtons per meter, you get a different curve here, where you get the correct order, where you have the 0.45 or 45% pre-strain here. It goes on top, it's on top of it.
Yeah.
Any questions?
I think that's it. If you just go to your conclusions, I guess. Yeah.
Yeah, I did show one of the small experiments I did where I took the elastic foundation off of the middle of the model, and then it didn't work very well. I didn't include that in the conclusions, but when I started this, that's one of the things that I wanted to see, is if you change the foundation of the tissue, it changes its reaction.
Yeah.
And that's proven in like all of the literature that I've looked at. If you have a different different substrate, yeah, if you have a different substrate, then you'll get differences in development. Like if you have the tissue on glass, that'll be develop into foam. And if you have the tissue on a loose elastic substrate of some sort, collagen perhaps, then it'll develop into adipose tissue or something. Like I have graphs for that. And the one thing that I'm hiding that I didn't want anybody to ask a question about was, how do the springs on the bottom of the model relate to the substrate elasticity? And I can't answer that because the elasticity is in newtons per meter squared, and my springs are in newtons per meter. So I don't know over what area these springs are acting. They're a point source in a model.
Right.
So I don't know. So yeah, so what would you like to know about my conclusions?
Yeah. That'd be good.
Well, okay, so I've proven that they could, my model can model tissue, and that the tissues are reliant on their attachments and also the values of their structure. And then my model, my 3-cell model, can handle very large stress and strain. And the other models out there, like the Ogden model, can only handle up to about 20% strain or deformation before it fails. And most tissues yield between, 50% and 60%.
Okay.
So this is a better model in that way for handling tissue.
So tissues are can like survive under much more strain than, say, like your typical.
Yeah, much more strain than, say, iron or steel, although there are some steels that apparently go up to 500%, but if you don't count the special steel, you can, like a lot of materials quit at about that 20% strain or before. But tissue lasts better. It's good for 50% strain usually, although that 1%, if you go back here, if you get 10% pre-strain on lung tissue, it fails at about 10% or 0.1 strain. And this is all engineering stress and strain here. This is an engineering department, and they were sure to tell me that this was engineering stress and engineering strain, but I don't want to emphasize that either because I read this paper and I think that's what they were doing, but they didn't say.
Oh.
Yeah, this one says that it's engineering stress and strain. This one, from reading it, yes, that's what they were doing, but it's like, I think some biologists wrote it, not some engineers. I need to know whether this is engineering stress or strain. Please, just say it.
So the difference is like, what is the difference between engineering and non-engineering stress and strain?
Well, when a tissue, like for instance, my model,
Yeah.
back up to my model to explain this. When my model is compressed, this middle part here that I'm measuring, this little triangle splays out, like it flattens when twisting it. And so if you were just doing true stress, you would take the enlarged area and use that to divide your force by to get your stress. But I'm just using the original size for engineering stress.
Okay.
And not how it enlarges as it goes. So there's true stress.
Right.
So
that make more sense for cells versus like.
Yeah, so part of maybe the curve thing that isn't going on with my model is simply that I'm using engineering stress. But that seems to be how most of my choice of tissue examples is. Like they've done engineering stress and strain with it. So I'm comparing the same thing.
Okay. All right, that looks good. Well, thanks for presenting that, and good luck with this presentation today. And
well, thank you for letting me talk about it. I just feel too nervous.
well, no, I think it's great. I think it's fascinating. Like figuring out kind of how to model tissues in this way, and yeah, I mean, I see. Yeah, I
Yeah,
see problems.
well, the professor of biosystems engineering who knows his mechanics is quite impressed with the model because you can completely flatten the model and it's still giving you some values. Mind you, I didn't put any yield values in it. You could to make it a more complete model. But yeah, you can really stretch the model out.
Yeah. All right. Yeah. So I don't know if Dick has any more questions, but he asked me to check my email, so I'll check my email and get back to him on that. But yeah. So yeah, if that's all we have, I guess we can go. It's at the top of the hour, so. But yeah, good luck, Susan, with the talk.
Yeah, I don't know whether I'll go to the talk. Oh, okay, there's a nematode paper.
Yeah.
Oh.
Yeah, it looks good. I don't know. Probably
Oh,
present it. Yeah, next meeting.
Well, I don't know if I'll go to the SLAM meeting this morning. There's a nice SLAM meeting. Or maybe I will. I don't know. Just like, this is too nervous.
Yeah, yeah.
Okay, did I stop sharing? No.
Yeah, go
There we go. It's better.
Yeah, this paper Dick pulled up, yeah, it looks pretty good. It's about asymmetric cell division and C. elegans. They do some work in C. elegans. They propose that nonlinear signal response dynamics between symmetry breaking PAR polarity, which is this mechanism in asymmetric cell division and asymmetric cell division modules, effectively insulate each individual module from variation arising in others. Convincing or hand waving? Well, yeah, going into the paper, I don't know. But it looks like the PAR polarity is this PAR protein, and they're arguing, I guess, that the dosage of the PAR protein is initiating this asymmetrical cell division from symmetrical cell divisions. Yeah. So I'll do this next week and present on it. And yeah, we'll see what it looks like, what they're getting at here.
Anyways, thank you again. Like, it's nice to present this to a somewhat knowledgeable audience, because then, all I'm getting from my son, for instance, is, what was that again? He was kind of interested because he's interested in mechanical engineering, but it's just, he's a mechanic. That's what he does.
Right.
So, pays more.
Yeah.
He didn't get a job. He lost his job. He lost a really good job. Not getting along with the supervisor. Boom, no job. Had a job in 3 days.
Wow.
3 days.
Yeah.
Unbelievable.
All right.
So,
Yeah.
the other mechanic in my life is my daughter's husband, and he went and got a job for $200,000 a year.
Wow. Yeah.
Because he's a mechanic.
Yeah.
I took the wrong profession, obviously. You need to be a mechanic these days. Anyways, okay.
All right. Thanks for presenting, Susan.
Okay.
All right.
Bye.
Talk to you later. Bye.
Bye.